Hello, listeners. Yamina here. Welcome to this episode of the Dr. GPCR podcast. Before we jump into this episode, I'd like to share a few things with you. The December and last Dr. GPCR newsletter of 2020 is now available. If you haven't subscribed yet, please visit drgpcr.com/newsletter. Also, don't forget to share the newsletter with your colleagues. We cannot wait to bring you brand new Dr. GPCR newsletter editions in 2021. Second, we will be taking a short break of the Dr. GPCR podcast in January 2021 as our family will be welcoming a baby boy. But rest assured, we are already working on bringing you brand new episodes, starting sometimes in February 2021 with an amazing lineup of guests. Also, stay tuned for a major announcement regarding the podcast. We're also proud to announce that we are pursuing consulting opportunities in the GPCR field. For help with your R&D project, please visit drgpcr.com/consulting or reach out by email at hello@drgpcr.com. And last but not least, we will be closing this first season of the Dr. GPCR podcast with a series of interviews with phenomenal female scientists in the field. We hope you'll enjoy it. So hello, everyone. I'm Dr. Yamina Bershish, founder of Dr. GPCR. Welcome to another episode of the Dr. GPCR podcast. Today, I have the pleasure of talking with Dr. Masha Niv. She is an associate professor and vice dean of research and development at the Hebrew University of Jerusalem. Hi, Masha.
Hello.
So good to see you and so good to have you here with us.
Thank you for doing this and for having me.
Of course. Thanks for taking the time. I know the time difference is about 7 hours between us, and I'm glad we were able to find the time to bring this together. So you're an associate professor. You're in Jerusalem. Can you tell us a little bit more about your career and how did you get into studying GPCRs?
So I always had this kind of strange relationship with, chemistry, biology, and other different topics. So when I was deciding what to study, I was thinking about psychology, literature, art, and maybe chemistry. And of course, I started chemistry. I'm saying of course because I was born in Russia. So the Soviet upbringing is very clear about, you study exact sciences and that's it. So I had many ideas of what to do, but I went with chemistry, and I loved it. But I also wanted to do something that's good for, something that developing therapies, new drugs, helping people, doing something good. But somehow I got hooked up on theoretical chemistry topics, and I actually went for a direct PhD in theoretical chemistry, but always having in mind that at some point I'll get to the biological question, which eventually happened.
Wow! And so, did you already know at a young age that you wanted to be? Did you have an idea on what you wanted to be at a young age, or this happened once you knew that you needed to do something tangible, something with numbers and formulas?
It's interesting. I think so. My father is a professor of physics, and my grandfather was a professor of applied math. So I think I kind of knew that I want to be a professor. Then during some stages of my studies, I was actually not so sure. So actually what happened when finishing my PhD, there were a lot of BioTech startup companies in Israel. And I actually, and because I wanted to get into biology, actually right after my PhD, I went to work for a startup biotech company working on kinases. Mm-hmm. And that's how I got into actually, designing drugs, which I always had in mind. Into biology and into all this kind of, excitement and euphoria of making something new and doing something that has the hope of helping people very fast. And that was where I actually did my switch into biology. And after that, but there was a startup. So, you have the highs and the lows, and it seems to work. And then all the market went down. And anyway, so after 2-3 years there, I actually decided to go for a postdoc. And that's, I went to Harel Weinstein's lab at Weill Medical College of Cornell. And that's where I got into GPCRs. So I actually was, so the steps from chemistry were going from chemistry into peptides, first of all, which often happens to a chemist because it's like the smallest, it's the smallest protein you can think that still looks like. So it was peptides that could inhibit kinases in the biotech I worked for, and then peptides or the C-terminal part of GPCRs that interact with PDZ domains, which I studied in Harel's lab. And of course, there in his lab, I also got into GPCRs more generally, like into the big, beautiful architecture of GPCRs.
Wow, so you went from a PhD in chemistry to a biotech working on peptides. And it's true, when you think about, biology, the smallest unit that you can think of from protein is a peptide. And then you went to do a postdoc, and that's when you got into the GPCRs in general. Where did you complete your PhD?
That was also the Hebrew University of Jerusalem. Okay. So, yeah.
You still had the opportunity to, work at the university, do a PhD, and then that's great.
Well, actually, you said in Jerusalem, but actually the faculty where I am working now, it is one of the faculties of the Hebrew University of Jerusalem, but actually it's located in a different city in Rehovot.
Oh.
So I'm not like in the same place where I was before. It's a different city, but it's still the same university, which I love. So it's all good.
That's awesome. So the university has different campuses. That's great.
Yeah, it's a challenge too, by the way, to have different, to have it dispersed. But yeah, but that's what we have. We have 4 campuses. Yeah.
Wow, all in different cities?
No, so humanities is one campus in Jerusalem. Then the medical school is a different campus in Jerusalem, and then the exact sciences and math, where I did most of my studies in yet another 3rd campus in Jerusalem, and the 4th one is in Rehovot, where I work now.
Wow.
So, yeah.
Wow, that is awesome. And so you mentioned that you went for a postdoc, and that's when you got into the world of GPCRs. Was there any particular moment when you said to yourself, well, I have to work on this. This is so cool?
During my postdoc, I guess, well, I mean, what happened is that the really exciting time was that the structures started to come out, right? So I started my PhD in 2003. And then when I got actually my position back at the Hebrew University, my independent position, and I had to choose the topic that will be like really my main topic for my lab, that's when I said, wow, the GPCRs, they are amazing. And now there are structures that are coming out. And that was really, the time that I knew that this is the topic I will want to stick with, even before the structures, they were great. But at this point, it was really not giving up on that. And then another interesting thing that happened to me is that the department where I work is the Institute of Biochemistry, Food and Nutrition. So I had also to pick a topic that would be relevant for for where I am, for my environment, and the other PIs that work with me. And there I really I think I made a really good decision to choose the taste GPCRs. So I decided on GPCRs, and then there were different options because, of course, they're involved, in metabolism and a lot of different things. But I think I chose it because there is a professor emeritus in the department who worked on taste before, and I thought, okay, it would be nice to, to have someone who is in the field, and this is amazing. And that's, I often say to people who, when new PIs join the faculty, they have to choose and what to focus on. And I often tell them that I felt that the maybe limitation or the fact that I had to choose a topic that would have a specific flavor, a specific actually was good for me, because that kind of made for me a niche which would be different from what my postdoc lab was doing or from other people are doing because of this like special situation. So actually I think there is an advantage in having something like that. And yeah, so
definitely. And taste receptors, I mean, I think we need to study them more. We don't know nearly enough about taste receptors. So not only you chose a great topic which fit with the institute that you were at, but also it's a topic that definitely needs more research. So tell us about taste receptors. Do you have a favorite taste receptor? What do they do?
Okay, so I have so first of all, I started from bitter taste.
Mm-hmm.
I think often talking about taste, you kind of always compare it to smell. So the olfactory receptors are also GPCRs, also very, very interesting. But the one of the differences is that, first of all, there are many more of them. There are like, for humans, like 400 different olfactory GPCRs, and many more people working on them also. But one of the questions, being a chemist and coming from this perspective, so one of the questions for bitter taste was for me similar to some of the questions in olfaction, which is the situation where you have many subtypes of the receptors that you have, and also many ligands, and how they, which ligand goes with which receptor, and it's not one-on-one, and does it mean anything that one ligand has many receptors and one receptor has many ligands, and how do they do it mechanistically? Like, how is it possible to actually to fit so many ligands into the same receptor? So the bitter taste, humans have 25 different subtypes of these GPCRs, and so it's a little easier, I guess, than the olfaction, which is really huge. But also this question was there, so I started really from that, and we started also, because I was a newcomer to the field, I would read papers, and a paper would say there are thousands of bitter molecules, and then the paper continues. And then I was like, okay, there are thousands of bitter molecules, but what are they? And so we first of all started from setting up the BitterDB, the database of bitter compounds. And then we started looking into how, into the associations, which ligands go with which receptors, and whether there are particular properties of the binding site, which is a predicted binding site based on the homology model, but anyway, of the binding site of the bitter taste receptor that would help predict or understand why some of these bitter taste receptors have many ligands, others have few, some are promiscuous, others are more specific, and still others are even orphan, and so there's still no ligand for them. And by the way, when we say orphan, actually maybe more of them are orphan in the sense that the bitter taste receptors and also other taste receptors are not only expressed on the tongue, they're expressed in different tissues in the body. So even though we know some ligands that are relevant to food or relevant to things coming from outside the body, it may very well be that in addition to that, there are some endogenous ligands for bitter taste receptors in the body that we still don't know. So maybe they're orphan in that sense, that we still don't know the endogenous ligands. So I like the family. I like that it's a family, that we need to understand what's going on, and we need to, so now we have a really cool project of matchmaking. So we're developing with colleagues, the data science at the Hebrew University, we're developing like a recommendation system where you have like different receptors and different ligands, and already some associations, and the properties of the ligands and of the receptors. And now we want to be able to suggest for any new ligand which of the receptors are suitable for them. So like you suggest a movie for a customer based on what you know about what similar customers liked and so on.
On previous reviews.
Sorry?
On previous reviews.
Yeah, exactly. Previous reviews of this one, and also like what he looks, who is his friends, who he went to school with and stuff like that. So, so the family aspect of this is something that I really like. But if we are looking at like a single receptor, my favorite one, I think I would choose the T2R14. This is one of the more promiscuous ones with many known ligands, and we've been working on this for a while because we want to know what else can activate it, we want to be able to inhibit it, and we want to be able to have very good ligands, because eventually we would like to solve also experimental structures, and for that you have to have really high affinity ligands to stabilize the receptors.
That's really great. So for bitter taste receptors, you said that they're not only expressed on the tongue. Do we know anything about the pattern of expression in the body? And also my next question would be, are these receptors expressed one receptor per cell or multiple receptors in the same cell?
Okay, cool, great questions. So first of all, for the bitter taste receptors, they're expressed in upper airways. Actually, they were suggested as new targets for asthma. They are also expressed, but they're also expressed like in the heart. Some very interesting involvement is in cancerous tissues. So it seems that sometimes there even might be a relation between being sensitivity towards drugs, depending on the levels of expression of these receptors in the cancerous cells, which could be related to perhaps their roles as identifying and helping activating pumps of pumping drugs out. So these are some of their interesting roles, and of course in the gut, they're also expressed in the gut. So I mean, very, very interesting. So regarding how they expressed in one cell, so in the tongue, in taste receptor cells, so the taste receptor cells either express bitter or sweet or umami, like the receptor or the channels for sour and salty, so they don't mix in one taste cell. However, you can have different subtypes, like of bitter, they can be expressed in the same taste cell. And this opens, a whole issue of the fact that they expressed in the same taste cell, kind of led to the notion bitter is bitter is bitter. It doesn't matter which bitter taste receptor, they can be together. So, bitter, that's it. That's like what they do. This is one school of thought. The other school of thought says, no, okay, they're different. And maybe, maybe in the taste cells, it's not so important exactly which kind of bitter taste receptor you activate, but they have other roles in other tissues, and in the other tissues there are different expressions of different subtypes. So I think they do, like, I think there are nuances and there are differences, at least in some of their roles, between the different subtypes.
Yeah, when you mentioned that you can have different subtypes of bitter receptors in the same cell, the first thought that I had in mind is maybe there is a scale of bitterness and you can detect it, or the cells at least detect it, depending on what subtypes may be co-expressed, or who knows. There is much more to do, definitely.
There is a lot to do, definitely, yeah.
So you mentioned that these receptors are expressed, in cancer cells. They can be expressed in the heart. It reminds me of a conversation I recently had with Antonella Di Pizio, who was in your lab, and we're talking about smell receptors, which is why the question of one receptor, one cell came up. And so they're also expressed in the gut. What would be the physiological role of a taste receptor expressed in the gut? What would it do?
Well, this part of it is known because they are related to gut motility. So basically, it's like, it's like one whole system that has to, you taste it, it announces what's going to happen. Then so they do have functional roles in the gut related also to secretions of hormones. So it's a feedback loop. It's not all clear, but they definitely have physiological roles as well. So I just want to say that my new favorite receptor is actually the sweet taste receptor. Which is also GPCR, but this is family C GPCR, so they're to the heterodimer.
Yeah.
So I started. I mean, this is really, really cool as well because, of course, it has very, applied things to do with it because you want to think about new sweeteners or even better sweetness enhancers, which would like make you feel the same sugar but feeling it sweeter, and then you don't need to have so much. But one of the first projects that we did with the sweet taste receptor is actually regarding the sweet taste of deuterated water, of heavy water.
Wow.
So Pavel Jungwirth, he's a professor in the Czech Republic, and he's a friend from the time that I was still in chemistry and PhD. And we met a few years ago, and he told me that deuterated water is being used in NMR experiments, and so NMR people kind of tend to sometimes taste it.
Okay.
And there was a paper from the 1930s, where Urey tasted it and said that it is not sweet. But since then, people were like tasting it here and there. So Pavel told me they're interested in that, and then since then we actually started working with this systematically, and we ran systematic sensory experiments on the taste of deuterated water, and we showed that indeed it is sweeter than regular water, and we were able to show that you need to have the heterodimer sweet taste receptor in order to elicit response in HEC cells. So we do, and lactazole, which is an inhibitor of the sweet taste receptor, inhibits the sweet taste of deuterated water. So this is like really strange because it's such a small change, and you get this effect. So we know that the sweet taste receptor is involved, and what exactly it is doing, this is ongoing.
Wow, it's you're in the taste, definitely in the taste world, deeply immersed in bitter and sweetness. But who would have thought that, such a little change in the water, you would make the body would make that difference. In sweetness, yeah, definitely. And I think when it comes to sweets, it's so interesting because if you eat sweets on a regular basis, at some point you get kind of desensitized, so you have the tendency of eating more and more and more. But then when you cut the sweets, which is hard to do, I'm sure everybody can relate to that, and then you come back and you eat something very sweet, it kind of hits you. You're like, oh my God, I couldn't taste this sweetness before. So I think there is a lot of nuances when it comes to sweetness as well. Wow. And do we know anything about the involvement of these these sweet tasting receptors in any pathological disease, in any diseases, or what do they do other than helping us taste sweet?
So they're also expressed in different tissues, and so their involvement is also in relation to diabetes still being studied. Mm-hmm. But one big thing that happened actually in the last month, of course, is this issue with COVID-19.
Yeah.
So approximately in March, reports started coming out about smell being impaired in COVID-19, and I was not going to work on COVID. I didn't want to get into that because I feel I never worked on viruses. I don't know. But then there was this report about taste as well. And I'm like, all right, if it's taste, what can I do? I have to I have to get into it. And so what happened is actually one really interesting, amazing experience for me, which is that like from a couple of tweets from people who were interested in that, we got into emails, then into Slack, and then very fast there was this huge GCCR community, the Chemosensory consortium community, with which I've been working for several months now to trying to really understand what's going on with the smell and taste in COVID-19. So because of the interest of my interest in taste, so we did ask the participants that we surveyed about their chemosensory losses during COVID-19. So we wanted to ask about specific tastes like sweet, bitter, in particular, each one separately, for two reasons. One, it's known that when we actually confuse taste and smell together. So it's kind of difficult to differentiate what is what, because together you have an integration of the senses, you have the flavor, and so the first tendency when people said the taste is impaired, that people would say, yeah, of course, your nose is blocked, you can't smell, you can't taste, that's it. So we really wanted to see whether reaction to non-volatile taste compounds, individuals ones, is also impaired. So we found that it is, and then on the other hand, we also found, and this is a paper that just came out yesterday on a medRxiv, in fact, with the GCCR, that even though both of these senses are impaired, strongly impaired in many or like 60+% of symptomatic COVID-19 patients, if you want to be able to predict if a person has COVID-19 based on the symptoms, it actually turns out that the quantitative smell change, so how much of your smell you lost, is the best predictor. And taste is not as good predictor. So it is a good predictor, but not as good. So that was funny because I really just got into it because of the taste, but it turns out that first of all the taste modalities usually in COVID-19 patients are impaired together. So it's not either one or the other, it's like together. So that actually is a hint that probably it's not the individual taste cell that is impaired, but probably other components that surround it, and then everything is impaired together. But the smell is the more telling symptom as far as we know so far. Yeah.
So if you were to ask which one wins, it'll be the smell and not the taste.
In this case, yes. However, there are many people who are anosmic or hyposmic or have actually previous olfactory disorders, not related to COVID-19. Okay. And that's where taste then comes in. And we actually had a really interesting case, a specific case of a person just like that. So if your smell is already bad, then you have to pay real attention to taste, because that can still be impaired even though smell is unchanged or is bad anyway from before.
Wow. Still, you answered my question before I was able to ask it, because I wanted to ask about, how did you get involved into studying taste receptors in the COVID-19 patients, and it's how interesting it is. The way I see it, when these patients show these symptoms, it's kind of the whole system gets shut down, and it's not just one taste or one receptor, it's just everything that gets completely dysregulated. It can be very interesting. With the one thought I had is, for example, alteration of taste in pregnancy, where the hormones just go overload, and sometimes there's things that you cannot eat while pregnant, and before you were able to eat that. Can you tell us, do you know anything about that?
So actually we had a interesting story with, so first of all, it is known, it is more common, I think, actually again in smell, that the smell is can be stronger, so hormone related I actually am not sure that I know about taste. It's in any case more rare, these disparities. But so we just had a participant who told us that she didn't realize that she had problems with her olfaction until she got pregnant. And then she started smelling better, and that's how she understood that before something was, because she always had some baseline that was like her normal, and then when she was pregnant, then she saw what's going on and how it can actually smell stronger, and then subsequently, every time she could smell well, she knew she's pregnant actually.
So no need.
But that's not a common pregnancy check, right? Usually you do something else. Yeah.
No need for a test. Her smell.
In her case, but usually you do have to have a different kind of test.
Yeah, that's still very intriguing. What are there any diseases related to any issues with taste, or the opposite? Are there any diseases that affect taste other than the COVID situation?
So first of all, a lot of medication. Okay. First of all, there are neurological diseases that are related to chemosensory, and especially smell, but also sometimes taste. But what is probably the most pronounced thing with taste is that you have a lot of medicines that are very bitter. Mm-hmm. This is one thing. And I'll come back to this in a second because we just did some work related to that. And then there are some medications, and in particular also radiation and chemotherapy that can impact the ability to taste. And this is a real concern because patients have a really problem with the taste. It tastes either really disgusting, or they can't taste and then they don't have appetite. So it's a big problem. And then, as I mentioned, something related to cancer. This is still very, very novel, but there could be issues related not to taste per se, but taste receptors that are expressed, like in cancers, and then could maybe help understand how to combat this disease. But one general thing is really is that many drugs are very bitter, and you can think about all these proverbs. If it's bitter, it will not cure you. The bitter pill, bitter. So we started looking into this from different perspectives. So first of all, in addition to this bitter in a drug thing, and in fact, traditional medicines sometimes, the traditional shamans and so on, they would use the taste to know, what is a potential drug. But on the other hand, bitterness is also usually considered as a marker of toxicity. So first of all, it's interesting because, of course, there is this relationship between efficacy and toxicity, and the window where it's efficacious but still not toxic, and it's all a matter of doses and so on. But what we did, we first of all actually wanted to know whether it is true that bitterness is a marker of toxicity, and we found that it's not such a great correlation. So you have many bitter molecules that are not toxic and many toxic molecules that are not bitter. Then we wanted to look into this issue with the bitter drugs, because it turns out this is really interesting actually, because in drug discovery, computational tools that are being used in drug discovery are so sophisticated, right? You have structure-based screening, and you have toxicology prediction, and you have, and of course, machine learning now and everything. However, this one aspect is not of the taste of the drug is actually not being considered until quite late stages in the development. So when do people look into it? Either when you have already to test the drug against a placebo. Now, if the drug is very bitter and the placebo is not very bitter, that's not, it's not exactly the same. So actually, when getting to placebo, you have to know if you have to like correct for the taste of the drug. It's one thing, but then usually actually what happens is when the drug gets already to the market, and then it turns out it's so bad that it can be, an amazing drug, but it will not be taken. Especially the problem is especially with pediatric patients, geriatric patients, and mental patients, where like swallowing could be a problem, as swallowing the big pills that are coated. And so now actually the FDA made it recently a requirement that the taste is reported. So like now it becomes something to take into account. So how is it taken into account again now? So there are sometimes animal studies with rodents that they drink less of the compound, of the water containing compound that is bitter. But what we have done is we have now developed a computational, so a machine learning predictor, which we trained on intensely bitter compounds and the ones that are not intensely bitter. And so now we can actually look at the structure and predict if it's going to be intensely bitter. And this is this actually has the potential of saving a lot of time, because this is a very fast tool. So you can take a huge library in the very early stages of development and already mark, this one is going to be bitter. Now, very bitter. Now, I don't want to say get rid of them. No, because I think actually many of them can be very good, because we know that bitterness but we can flag them and we can say, okay, this one might have issues, so let's keep in mind and think about formulations and think about testing. So so this is one aspect of actually any drug for any disease which you have to swallow, that you have to think about bitterness. And then of course, like the other part of it is because we have bitter taste receptors in the intestinal tract or in different tissues. Actually, probably some of the drugs that we are already taking for another target, some of them might be already having an effect on the bitter taste receptors in our body, and maybe part of their effects already have to do with that, even though that was not the purpose. But yeah.
Wow. So you definitely there's potential and a lot of work to be done to better understand how taste receptors and bitter taste receptors actually work. And I haven't thought about the formulation, but you're right. Whenever you take a drug that is really bitter, and it's a huge pill, it is a problem, and you want to just swallow it as soon as possible, because it does leave that aftertaste, which can be a problem with a child or with someone.
Yeah, and when I was a young child, actually, I had really huge problems swallowing. I mean, it was difficult for me, so I can really relate to that.
Yeah, and then I think I remember times when the pills were crushed so that you can swallow them better, but then you release the whole taste of it, and it's even worse.
Right, exactly. Or if you're trying to chew, if you can,
Definitely. So it's not ideal, but that's great. So you have this kind of a machine learning tool that in the beginning already gives you a kind of a scale of bitterness that can be taken into account on whenever you have these drugs or these compounds that are being used. Are there any ways of, for example, you're developing a drug and they know that it's extremely bitter? Are there any ways of making it less bitter but still keeping the activity, or make it sweet or something like that?
Yeah, so there are different approaches, of course, of like cyclodextrin encapsulations. There are different approaches. There's still, there's still need for additional ones. Mm-hmm. And it's a big topic of study, actually, of how to, like, if you basically you're saying, okay, so you found out it's going to be extremely bitter, what is the next step? Exactly. So from my point, I mean, there are different approaches, many like physical mechanistic approaches, but from our point of view, what we are trying to do is develop antagonists for bitter taste receptors, because if this is not the target that we're targeting, if it's just an off target, maybe we can do something that would not activate them or inhibit them. This is an interesting thing about bitter taste GPCRs. It's more difficult to find antagonists for them than to find agonists for them. So we've been looking for agonists for a while, and for ligands for a while, and we are very successful in finding new agonists. And for antagonists, we have successes, but this is what we are trying to work on.
Is it because people have been from the beginning more focused on finding agonists, so they didn't kind of take the time to look for, or is it a special property of taste receptors?
I think it's a property of these receptors. They are more prone to getting activated. There are particular subgroup of family A GPCRs. They're missing some of the conserved motifs, some of the disulfide bridges. They're not exactly like all the other GPCRs, and I think it is something to do with their architecture that makes them, easy. It's easier to activate them.
Interesting. Well, when you think about it, since it's a taste receptor, when we just consider the tongue, at that point, evolutionarily speaking, you really want to detect that bitterness taste. So you really want to make sure that you activate it before you eat that fruit that might kill you.
Exactly. Absolutely. Yeah. In terms of this physiological role, it makes a lot of sense that that's what it should be doing. Yeah, like sensing and going, oh yeah, there is something. Yeah.
Exactly. Maybe I shouldn't be eating this. Yeah. We've talked about this, and the answer to this question is always yes. But I was hoping you can elaborate. So, do you think GPCRs and taste GPCRs could be good drug targets in the context that we spoke about?
The bitter taste, I believe, yes. I believe that they will turn out as drug targets. The others, maybe also, I'm not sure, but I'm pretty sure that the bitter taste, yeah, I think so.
So you had mentioned that there might be an application in asthma for the bitter taste. Were there any other applications that you can think of as drug targets for the bitter taste receptors? Well, cancer as well. You were mentioning that sometimes the cancer cells also express.
The cancer is still not like a mainstream thing. This is there are some works from Matt Gaida from Germany that he showed this difference in pancreatic cancer in patients who express or do not express these receptors. So I'm just mentioning it because we are collaborating with him, and I think it's a very, very interesting and promising direction, but it's very novel. It's not like a main thing that everybody kind of agrees on. I think also, I would say that asthma probably is the most advanced, like it's the first application that was suggested. But because of their expression, actually, in fact, I think also heart or like cardiology related issues might come up as well, because they're also involved in I'm forgetting now how it's called, but like heart regulation of heartbeating, I think. Yeah, so there is still work to be done. Yeah.
Yeah, I think we were looking for cardiac arrhythmia.
Right? It's called ionopathy, I think. Anyway, yeah, I'm not sure, but it has involvement. It was shown in mice hearts, and now, but it's not yet. Probably it is not the like the number one next target, except maybe for asthma. It's been debated much more than other things.
Wow. Yeah, I can see a lot of applications, not only in disease areas, but also in food, as you were mentioning, to develop molecules that taste sweet, but you don't need to eat the entire bag of cookies.
But In food, though, it's interesting because in food, we kind of have this tendency of, getting rid of the bitter, and of course, sweet is very attractive, but I think there is some work to be done in terms of like nutrition and set of mind, of kind of accepting bitterness in a more open way than is usually done in the Western, diet. Because first of all, in Eastern diets, the way they treat bitterness is different. We have this like, you accept maybe for beer and coffee, everything else, is not. But in actually bitter compounds, first of all, we found that they are not necessarily toxic, right? I mean, there are some families, but not usually. And there are a lot of very healthy compounds that have slight bitter taste. So actually, in a way, and this is something my colleague Angela Basoli is really promoting, is like the bitter for better health. So let's not think about bitter as something like just to get rid of, or to try to put like a lot of sugar on top of it not to feel it, and rather instead try to get used to it and to accept it as something that is actually not bad, and that would actually be a healthier diet for us if we accept it. And
so let's all accept bitter taste.
Some bitterness, yeah.
yeah. But of course, I mean, can you think of any compounds or of any foods that are bitter but still are healthy? Now you mentioned coffee and beer, but what are
a lot of flavonoids, a lot of even caffeine and other compounds in coffee, humulons from beer. Are bitter and healthy, but really flavonoids from citrus fruits, from other fruits, compounds from tea that are bitter but also antioxidants. It's really a diverse group of compounds that has a lot of good in them.
Yeah, so we shouldn't be afraid of exploring the bitter tastes. They can be healthy, which I think it's I think the more varied you can think of your nutrition, and then you get more nutrients in there, more vegetables, more and that's the goal of getting all those good nutrients in, and not stick to the sugary
exactly thing. Exactly. There are really beautiful works of Julie Manella and others where they're trying to get very young children used to both the variety and also, the particular tastes. And so in some cases, there were even works trying to do it through mother's diet while she's still pregnant, and then breastfeeding to really, because early exposure and repetitive exposure to flavors has a major effect on what is later on accepted and liked.
Oh, I agree, and I think that's a whole other topic for another podcast. But I think that's also very important from a very from the pregnancy to make sure that you choose different foods, and then also give that choice. And I think also when you look at it, so I think you eat with your nose, you eat with your tongue, and you taste, but also you eat with your eyes. So having that diversity in color can also be a very, very appealing to everyone when you look at different foods.
This is, of course, absolutely true, but I just have to mention just one very cool collaboration that we recently had with Craig Montel, where he showed, and actually it was together with Antonella when she was still in the lab, that the opsin receptor in Drosophila is actually also a bitter taste receptor, in the sense that it can be activated by bitter compounds.
That is
this is a really, it's really cool work where, maybe kind of tells you even the origins, like maybe the visual receptors originally or very, very early on in evolution were in fact chemical receptors for some chemicals, and later on, with the retinal that could be activated by light, then it became a vision receptor. So anyway, I know you meant something else that you see and it affects you, which is completely true. But in this particular case, also there is this, combination on the chemical or the receptor level.
That is so cool. So yeah, with the Drosophila, you also can again taste with your eyes, kind of, which is amazing. So there is a lot of work to be done in the field. There's many applications to studying taste receptors and bitter taste receptors as well. What would be your advice to junior scientists who wanted to contribute in the field or wanted to better understand these fascinating GPCRs?
So I actually, first of all, I think my advice to scientists is to do something that I have done recently, but I think now everybody should do it, is go on Twitter. Okay, because the amount of information, of opportunities, of the speed with which you get exposed to results, and also a feeling that you get from like different labs and what are the people behind the science and what they care about, the inclusiveness and diversity, and there are other values that you will kind of see from what they're tweeting. So it's really amazing. So for me, from Twitter, I got to establish and to know the amazing people in the GPCR consortium. It also helped us establish GPCR Ladies website, which is a website which lists the women working in the GPCR field. So if women listening and wanting to be, there is a link where you can put your data. So we added, and many other things. Good things came from me on Twitter, even though it's difficult because you can see like everybody's so active and so many things are going on. It's also a challenge in its own, but nevertheless, I think it's worth the time to follow, people you appreciate in the field, and to get exposed to the numerous free resources and virtual conferences and so many things that are now going on.
Oh, definitely. I mean, when I started the podcast and started working on Dr. GPCR, one of my first questions was, where are the GPCR folks? Where is everybody? And it turns out everybody is on Twitter. And in spite of the whole COVID pandemic that we're going through, there is such an enormous amount of activity in the field that's been going on. And it's so refreshing to see that people are still continuing to work and there's papers coming out. We have a monthly newsletter that comes out, so people can subscribe to it, drgpcr.com/newsletter. And we compiled last month only 10 papers only quote unquote 10 papers on structure. But I think at the end of the month, we had about 60 publications that were in some way related to GPCRs. Most of them had a focus on a specific family or a specific receptor, which I think is phenomenal. To be able to just, scroll through those and kind of cherry pick what you're interested in, but also look at articles that you normally wouldn't look for or look at. Just having that diversity, I think, is just phenomenal.
Definitely. I agree. And I don't know if we have time, but I actually wanted to ask you a question. Can we do it?
Of course.
So I was really impressed by how you went, I don't know, like from there was no podcast and now it's like, wow. And so I wanted to ask you whether, like, is it something, how did you decide to do it and how do you learn to do it? Like, how did it happen?
So it happened sometimes in February, where I was listening to a podcast about consulting called Consulting Success. And they're Canadians as well. So, and they were talking about how to kind of establish yourself in the field and how to create credibility. And a couple of years ago, the answer to that was write a blog. And although I do enjoy scientific writing, I felt like it wasn't up to date. It's 2020, come on. And I figured, you know what, let's just talk to people and create a podcast. And I knew nothing about how to create a podcast back in February. But I did what you'd call a market research. I did talk to people who might be coming on the podcast, and that's how it went. And I really very much enjoyed. I enjoy talking to people like you. We're opening up the floor to trainees to postdocs and PhD students, obviously with a different set of questions, but the idea is to also promote all of us who work on GPCR. So that's how it came up.
That's very cool.
Yeah, thank you. And it's been a great adventure so far, and it allowed me to talk to people from all around the world, which I think that's kind of the diversity that we want to have in the field. Independently on where you are, what your time zone is, if you have an interest in the field, then we should all belong under the same umbrella.
Absolutely.
Super. So my last question to you, Masha, is when you have job openings in your group, where do you usually advertise? And I wanted also to let you know that we do have a career page. There's drgpcr.com/career, where we're also gathering all of the GPCR related job postings independently, whether they're postdoc or a PhD or even industry jobs.
Thank you. So, so for me, I think, how do I do it? So if somebody wants to work with me, so probably Twitter is a good place to look. Then I advertised through costs that I belong to. So like Gleason, and now it's Ernest.
Yeah.
That I know some people through Ernest, they're coming also to talk here. And then I guess these would be the main places I would advertise. Now there's also this job archive repository, which I have not used. It's like, like medRxiv, bioRxiv, there's job archive. It seems, so I see people advertising there and then showing on Twitter what they advertise. So that seems to be like something to look into. But definitely, I will look also into your advertisement now. Right now, my lab is pretty big, so I'm good. I'm good with students. But definitely in the future, or if people come with their own funding, that's perfect, and we're open.
That is awesome. That is amazing. There's one last thing I wanted to say, but it kind of flew. Oh yes, so we were talking about Ernest, and I think it's a great opportunity, for all of us in the US or anywhere in the world, and with the European team as well, to come and get together and kind of share all of these resources. So I think it's a good time to be working together, and not having to travel or not being able to travel also has its advantages.
Absolutely, yeah. I just had an idea that, okay, I'll tell you after we close. My idea. Okay.
All right. Well, we are at the end of our chat. Thank you so much, Masha, again for your time, and talk to you soon.
Okay. Thank you.
Bye.
Thank you for listening to this Dr. GPCR podcast episode. We hope you enjoyed it. Subscribe to the podcast wherever you listen to podcasts. I'd like to thank our guest, Attila Forrest, Jin Chong, and Shivani Sachdev. Music by Rosa Bershish. I'm your host, Dr. Yamina Bershish. We're always excited to hear from you. Visit us at drgpcr.com or send us a note at hello@drgpcr.com. I also wanted to wish you happy holidays and a very happy new year. I'd like to thank this year's guests and thank you for being such a wonderful audience. Thank you again for the privilege of your time, and until next time, stay safe.